Anyway, since the session is going to start, I request the chairman, Dr. A. Latha. She is the Director of Research of the Kerala Agricultural University to occupy the chair. And I also invite Dr. Anita Karun, Research Institute also to join her. And I also called Vasudevan sir. I think this.
Vasudevan sir alla.
Venugopal. Sir, please come. Here. Okay. Then also Murli ji. He will be.
Okay, I'll say something. Messages. One from Professor Hiroisa Tamagawa. He's basically a mechanical engineer who works with polymers, and he also has another dimension. He works on the electro and neurophysiology. So he has been collaborating with me for the last 5 years. He would like to share some message. He was supposed to come online, but his mother met with an accident. So, yes, please, Professor Hirosa Tamagawa.
My attention. Therefore, I regret that I am unable to attend this meeting. I sincerely apologize for my absence. I would like to briefly show my impression to the murburn concept. The murburn concept is an entirely new physiological theory because it represents a fundamentally different way of thinking about physiology. Many researchers understandably hesitant to accept it. However, I believe that many scientists have already recognized the limitation of existing physiological theories. At the same time, I understand that it is not easy to move beyond the long-established ideas of physiology and embrace new frameworks such as the murburn concept. I sincerely hope that the murburn concept will open the door to a new era in physiology. Thank you.
So, Markov, here is PowerPoint, please. Next is Dr. Marco Fiore, who is an Italian researcher from CNRS. His family, 2 people have gone away for a holiday in Alps, so that's why they are not able to join. So here he is.
From the Institute of Biochemistry and Cell Biology of Rome in Italy.
Next.
We're going to speak about the murburn concept. The question is, could the murburn concept reframe mitochondrial oxidative processes to frame murburn concept as an intriguing oxygen-centered hypothesis?
Next.
We are going to interpret as that mitochondria as controlled oxidative species. Mitochondria are not just ATP factories, they are structured oxidative microenvironments where electron flow, oxygen reduction, loss generation, proton gradients, and the membrane architecture coexist. If ROS are not present in mitochondrial respiration, are they merely accidental byproducts or are they part of the operating logic of the organelle? And now, what is the murburn concept? As far as I know, the murburn concept proposed that diffusible reactive species are not just toxic leakage products, but functional participants in redox catalysis. The question is, could mitochondrial membranes act not only as proton barriers, but also as radical reaction boundaries that confine and regulate oxidative chemistry? Indeed, the murburn refers to mild container redox chemistry involved with disposable reactive species. Murburn proposes that ROS, RNS, and other diffusible radicals can mediate catalytic processes outside the classical enzyme active site model. In mitochondrial oxidative phosphorylation, the Murburn model proposes an oxygen-centered radical-mediated explanation rather than a purely proton-centric chemiosmotic one. This should be considered as an alternative and debated framework and not the standard consensus model. We are approaching the core of this business, Murburn and mitochondrial membrane stability. The key question is whether or not controlled ROS chemistry could help maintain membrane function while uncontrolled ROS causes damage. In oxidative stress, controlled ROS may support redox signaling and metabolic adaptation. Excessive ROS can damage proteins, lipids, and membranes. The Murburn view would interpret mitochondrial ROS as potentially functionally when spatially and kinetically controlled. In the membrane composition, crystal geometry and respiratory complex organization may determine whether or not ROS are useful signal or destructive agents. In the Murburn concept, what determines the switch between beneficial ROS-mediated regulation and destructive oxidative membrane damage? Crystal corpuscles and selected oxidation. Crystals are indeed good structure for asking whether or not subtle redox chemistry shapes mitochondrial function. Crystals are specialized in mitochondrial membrane domains containing respiratory complexes and ATP synthase. Crystal junctions can restrict diffusion and create local electrochemical differences. ATP synthase dimers help shape crystal curvature and organization. Murburn raises the possibility that crystals also help localize radical-mediated oxidative reaction. I had so many questions. For example, could the mitochondrial crystal be understood as organized mirror zones where membrane architecture controls the balance between energy production, redox signaling, and oxidative damage? In conclusion for this very short presentation, the Murburn concept does not simply ask whether or not mitochondria produce ROS. It asks whether or not controlled radical chemistry is part of how mitochondria work and act. The critical experimental challenge is to distinguish regulated oxidative microchemistry from accidental oxidative stress. And thank you so much for your patience.
All right, I think
No problem. So just testing the question. So I can wait for the photosynthesis lecture.
Yes,
Number one, Professor.
yes, thank you. So, then there are multiple aspects that we will cover in this. We do the most important reaction on planet Earth, which Indian Council for Agricultural Research stands fundamentally. Of course, here it is marine fishery research. Photosynthesis. All right, it gives us the food that we eat, the oxygen that we breathe, primary source of carbon-centered life that we see on planet Earth. So, most of you know from your school or higher secondary education, you have the chloroplast is a place where photosynthesis occurs, like mitochondria is the place where respiration occurs. So there are grana, there are stroma, structures like this, stacks of lipid-based membranes on which photosynthetic proteins are spread. Now there is a lot of information available where you know what kind of proteins are available more, where, which part of this grana, which are greater distribution densities there. So this is the ultimate fixer of carbon and the right reaction to ATP. This is what my concern. I'm not involved in Calvin side. We are dealing with how NADPH, the reducing equivalents, and ATP, the energy currency, and oxygen are produced. Photosynthesis, the day reaction, 3 fundamental processes. The reducing equivalents, the phosphorylated energy, and the molecule of oxygen. This is produced from water. Currently, the mechanism is highly deterministic. We'll go. All of it starts from the experiment of Emerson, a very famous experiment. Emerson experiment. What was done was oxygen evolution was seen. There were, at that time it was known that there were 2 types of photosystems. One that primarily gets excited at 680 nanometer, another at 700 nanometers. So they first excited at 680 nanometer, they got some amount of oxygen. Then they could excite it at 700 nanometer, some amount of oxygen was derived. The interesting thing was, when the system was excited at both 680 plus 700, the amount of oxygen that you would get, it was more than the combined of these 2. So, what those scientists did is, they said this is the stoichiometry, that is the ratio of the overall reaction, wherein first water was broken down at photosynthetic system 2, PS2, 680 nanometer, and then the electrons were relayed serially through some intermediary proteins and small molecules like plastoquinone and plastocyan. This is a protein, this is a membrane embedded quinone. So then finally coming at P700, that is for system 1, to reduce NADP to NADPH. So this is a serial electrons transport chain. It was called Z scheme. Why Z? Because it goes like this. So then the ratio was 2 molecules of NADP plus held at reductase, 4 electrons from 2 molecules of water held at PS2 to release 1 molecule of oxygen. That is, 2 molecules of water were held here to release 1 molecule of oxygen. Right? Now, the problem is, this is something that violates basic awareness of current flow, or basically traffic. You imagine there's a reservoir of electrons for system 2, for system 1. If you connect them in series, what will happen to the electron flow? Basic 10th class understanding of electronic circuits. When you connect 2 resistances in series, what happens to the overall resistance? It increases or decreases? Let's have some interaction. It's already afternoon. Resistance increases, so current flow will decrease. Is this a good idea to connect them in series? So now, suppose if I had to get greater traffic from here to, say, Madam, would I make one long road or multiple parallel roads? If I wanted to send more traffic, I would have multiple parallel roads. So it was a very wrong idea in terms of either fluid flow, traffic concentration, or even electronics or fundamental electrical connections. It is not a good idea to connect in series, it's completely wrong. So to explain Emerson experiment, they connected photosystem 1 and 2 in series. Nobody on this planet Earth can ratify this rationale. Govindji, who wrote the textbook on photosynthesis, his responses are officially noted on my YouTube website. Alright, so this is the general idea. This is how it looks like. Proton pumping. So the whole point is when chloroplast size excluded volume, there's hardly any protons. So how will it pump out? And the electronic circuitry goes down, up, out, all over the place. There's no directionality. How? So cyanobacteria can both respire and photosynthesize. This argument I had said earlier. So, and I also talked about the commonality of certain agents. How will it know what to participate in? Alright? And we also know that a membrane cannot just pump ions both directionally, different kinds. There is hardly any logic. So the idea that I wanted to very clearly say is whatever is given in textbook, you check out. See, this is 4 photons here, 680 nanometers, 4 photons here, 700 nanometers. So that's supposed to give this. You put that equation and you check it out. It requires 1464 kilojoules per mole. 4 photons each of 680 nanometers and 700 nanometers only give 1387 kilojoules per mole. So I don't know what they did to get the, even if you accept proton pumping as a energy conserving gradient forming mechanism, this reaction is unviable even in the basic treatment. Then deterministic key carriers. If you see the overall system, there are multiple. This is a quinone. This is a membrane quinone. I had shown its structure yesterday. Plastoquinone is a little different from coenzyme Q, but they are still quinones. Large lipids that span the membrane. It won't move like that. It has no way to deterministically move. And then this is a soluble molecule. How do they move like that with an errand? And particularly now that we know the distribution, photosystem 2 is deep inside the grana, photosystem 1 is outside, cytochrome B6F is outside. So the plastoquinone should jump thylakoids. How does it happen? Alright, so how does the proton pump here comes to cause complex Y that is here? It doesn't. This kind of setups, the architecture doesn't agree. And the very important thing is plastocyanin, its topology, and he explored this. He shows that, you see, if the electron transfer, if plastocyanin takes the electron from photosystem cytochrome B6F to photosystem 1, like cytochrome C takes from complex 3 to complex 4, similar parallel here. You would imagine that it would be conserved, its topology would be conserved if the binding based there is no conservation. And then plastocyanin is available at very low concentration, 1 micromolar. If it is so important, you should have it at higher concentration. Most importantly, they made in Arabidopsis thaliana, they knocked out plastocyanin completely, complete knockout, no plastocyanin, yet it works beautifully. So the plastocyanin or cytochrome C here, this is cyanobacterial. Plastocyanin, this part was knocked out. Sorry, I shouldn't have shown the respiratory chain, plastocyanin here in class. Then, if you see the structures of photosystems and the light harvesting complexes, the the colored pigments are scattered all over the place. And there is hardly any covalent tagging or tethering. So they just occupy random positions, it looks like. There are even colored carotenoids, even in cytochrome B6F, which doesn't have any, photoactive responsibilities. So why or how? And then the action spectrum also, there are, doesn't really, there are so many, there is decent amount of photosynthesis, not that efficient, even in non-PS1, PS2 regions. So then, here comes the murburn model. Rather than proton pumping and all that, here what we say is light hits chlorophylls and carotenoids and xanthophylls and all those pigments and all those, they can excite and liberate electrons which can be collected by NADPH and oxygen. So the most important thing is this is a highly decentralized parallel system compared to the serial mechanism and sequential serially ordered. Here everything works independently. There is no net necessity for that. What this scheme advocates. So here cytochrome B6F, ADP binding, all those membrane extra subtensions, Abhinav showed that they bind ADP. So what happens is here reactive species are formed. Reactive species can attack phosphate or the electrons can go into NADPH, and when it goes to phosphate attacking, it forms ATP. It's a random delocalized thing. But then somebody would ask, why photosystems? They are effective charge separating agents, quite unlike light harvesting complexes. So let's see, that's the one view. Here I would say there are two kinds of reactive diffusible species produced. One that is required for the engine proper and another for lubrication. Like for light harvesting complexes, they produce reactive species which help function at high photosynthetic times when electrons get displaced, replenish that. But they are not good at effective charge separation. So photosystem 1 and 2 are for that. So this is yet another view. Here is a more temporal kind of view. What happens is diverse pigments photoactivated, you get effective charge separation and electron emission, giving us diffusible reactive species. This diffusible reactive species can give electrons to photosystem 2 and 1 again, and diverse pigments, they give it back, and then they can also react among each other to form water and oxygen also. And photosystem 2 can better use H2O because it has got a manganese cluster which can afford better charge conservation. It can deplete peroxide also better, thereby liberating oxygen from there. So here diverse pigments can also electron replenish with this. So DRS is formed, it can replenish electron holes. So tertiary processes, membrane proteins can use electrons to generate DRS. They can give to NADP to form NADPH and also get phosphorylation. So NADPH synthesis, oxygenesis, phosphorylation, transmembrane potential generation, thermogenesis, homeostasis, all mediated through DRS in the murburn model. So the beauty of this model is that it is thermodynamically, mechanistically, kinetically justified. You see how oxygen evolution, it is not centered only on photosystem 2. If you see, the experimental observation doesn't mandate that only photosystem 1 can. See, Emerson's experiment clearly shows photosystem 1 is excited, it also produces oxygen. So that is agreeable to us, the murburn model. So here, what is saying, oxygen evolution is decentralized. All right, when the manganese complex, the OEC, so-called water splitting complex, that can effectively deplete peroxide and therefore tug the thermodynamic pull. It can drive the reaction. So that, because otherwise the 2 electron formed of H2O2 will linger and that will react with molecules and then it will not proceed further to oxygen formation. Alright, so you can see in many ways you can get oxygen formation. So a product formation is not by one route. It's like you have rainfall on the mountain, the water falls through multiple routes to come and assemble at a lake in the valley. So something like, there are multiple ways, multiple parallel ways in which oxygen can be formed. Similarly, NADPH can be formed, ATP can be formed in multiple ways. And all their energy terms are very simple, highly hexagonic. You can have ATP synthesis associated with oxygen evolution. So here you can easily get much lesser energy required for if you use hydroxide ion. Here you don't need 2 water molecules to be bound at one time. No such fastidious requirements. So in vitro oxygen superoxide DRS equilibriums mediate ET, electron transfer. We have shown through our 1.5 decades research, not one experiment, not 2 experiments, multitudes of publications we have shown. And then we showed also ADP plus phosphate in the presence of superoxide gives ATP, which is inhibited by cyanide. This was shown by Tiskiewicz and Rao. They, in 1987, they showed they took chloroplast, xanthine oxidase and xanthine, they got higher ATP, which is inhibited by SOD. So clearly all the experiments that need to justify the basic foundation of phosphorylation in the murburn model in the site chloroplast is available. Now let's see the structure of proteins, the thermodynamic, kinetic. See, these molecules are all fundamentally bimolecular reaction, though I have written it like that. First this attacks this and then something is formed, and then that attacks another one and this is formed, and then the 2 radicals in that react together to form the 3rd one. So they are all fundamentally bimolecular reactions whose reactions are facilitated by these kind of structures. This is photosystem 2, then this is NADH, NAPH dehydrogenase, this is photosystem 1. So you see, like I said, random scattering of chlorophylls here and there. We see the porous structures. If there are really beautiful electron transporting route, you will not expect such structures. And this is cytochrome B6F. It has carotenes here and there. There is no rationale why you should have subtending pendulous parts like this. Abhinav showed that they are, see here, ADP bind sites. Same case with NADH dehydrogenase, NADPH dehydrogenase. So all of this, the random distribution, the subtending, outside membrane subtending, redox centers, all that agree with the murburn model. So they are low affinity binding sites. Pigments and Ross channels are noted in proteins. The latter is inexplicable if the deterministic electron circuitry were physiologically accurate. Very importantly, through 30 years of research, many people have shown that very diverse types of electron donors can function for both PS1 and PS2, photosystem 1 and photosystem 2. See, all these have been demonstrated to serve as electron donors. Electron donors and acceptors for PS1 and 2. Then E acceptors from PS1 and 2. So, there is no real specificity for anyone to argue. Very clearly, there is no specificity whatsoever. And also, we had published with Professor Wu this fundamental major problem in photosynthetic chemistry. It was seen that bicarbonate, when you give to this system, it enhances the photosynthetic rate and oxygen release. So we showed that through the calculations that yes, it is somewhere between the values of energetic yields between the water, the amount of requirement for water or for hydroxide ion. So it can definitely, in murburn, it doesn't require specific binding, so this can also work. So diversity of E donors and acceptors from PS1 and PS2 and all that discredit the Z scheme. You don't need affinity bound moieties to give. Bicarbonate known to enhance oxygenesis, and Emerson effect supports decentralized oxygenesis. These are the two fundamental experiments that were misinterpreted for and which gave a misdirection to bioenergetic research. First is Jagendorf research. This somebody talked about yesterday. First it was done in chloroplast. They give pH 4, they equilibrated there, and then transferred to pH 8. In absence, they just added ADP, PI, and you could get ATP. So that means that a pH gradient works in chloroplast. Now, if you see the murburn equations for that, it will tell you why. You see, plus this gives NADPH. Everywhere where it requires H plus on the left side, you give, it drives the reaction forward. Alright? Even in the other one, ADP plus, PI plus, H plus, that phosphorylation also goes. Even this phosphorylation. In all these reactions, H plus can be a reactant. So if you give H plus from external source in a proton limited area, then you will get reaction. So that was a reaction experiment which had zero relevance to physiology. Zero relevance. Absolutely zero relevance. Then another experiment that was done was called the Racker's experiment. Alright? The Racker's experiment, what they did is, Ephraim Racker, he constituted vesicles and he added these different systems. That is, in one he put only bacterial rhodopsin. In the other he put only ATP synthase. In one he put both rhodopsin and ATP synthase. And in the other one he put rhodopsin, ATP synthase and an uncoupling agent. Now what was the observation? They said no ATP generated in bacterial rhodopsin, no ATP generated in ATP synthase, but ATP was pretty decently generated in rhodopsin plus complex 5, that is ATP synthase, but ATP synthase was, ATP was not generated with uncoupler. So they argued that uncoupler basically dissipated the proton gradient. Now here is the biggest problem. Without complex 5, this gave 20% of the ATP that was generated in this system. So they conveniently oversighted that. Because they said complex 5 with this thing, if this gave 100 molecules of ATP, this gave 20. And this also gave some, but it gave lower than this. So, disregarding observations, superposing one's own whims and fancies does not. This reaction, if you say complex 5 is the ATP synthase, why am I not getting in this? What they say that this works as a proton pump creates a gradient and then that comes back. What? It's a vesicle. So the point is, here you could see ATP synthesis without ATP synthase, so-called ATP synthase. That is the key observation. So they said this pumps proton. It's a very well known thing that rhodopsin's Schiff's base pK is around 13. There is no way this can function. It is 5 pH units above physiological pH of around 8 or 7, 6. It will not function as a proton pump. It cannot function. So these experiments had zero relevance to physiology, or they were misinterpreted. So this is the basic understanding. I'm not going into photosynthesis in greater detail. Fundamentally, we have shown that there is hardly any reason. Recently, Frontiers invited me to submit a paper, and they just refused publication prospect. Outright, they rejected everything poor. I gave them a peace of mind. I said, we have already published this. And then when I told them like that, 2 new rivers came in, and they kind of said, this is a great book. So sometimes you have to put your foot down when you know you are confident. All right. So this is the photosynthetic work. The next slide, please. I have a lot of other bicarbonate, all that. Other slide. We'll go over it quickly. Now 3:30. I wanted to give some messages to kids also. Yeah. So here, the other one you see, when our sir, Krishnamar sir said, ions, can they move that quickly? If I say, sometimes in Tibet, please listen to this example. Sukumar sir doesn't like me giving such examples, but this is how I can get your attention. Yeah. Sorry. Sukumar sir doesn't like me, he was saying, yeah. So I wanted to just take a break and we'll get back to the elephants are found in Tibet. There are no elephants naturally found in Tibet. So now you have, imagine, sometimes when the elephants are found, sometimes a red Maruti is also found, Maruti 800. It has 4 doors, 4 seats. Now, sometimes also those elephants also have Kunyiraman, Appakutan name tags in Malayalam. Alright? So, sometimes rain comes when the elephants are found there. So, the elephants, they are noted here, and within 2 hours they are noted in Tibet. Now, along comes somebody and they have theory. The 4 elephants sit on the 4 seats. They drive down from Kerala all the way to Tibet in that Maruti sedan. Will you buy it? Will you buy it, kiddos? Will you buy it? Why? Why won't you? There is 4 seats. There are 4 doors, elephants, correlations. When the elephants are found, the red Maruti is found there. It has Maruti has KL registration. KL registration. Why won't you buy it? You won't buy it because your ideas, day-to-day ideas of space, time, speed, abilities don't agree with it, right? Similarly, all these molecules like oxygen, they just can't bind and stay at one place. 2 molecules of water, without releasing oxygen, they have to be brought together. Oxygen should not be going anywhere. All this is not possible. Alright, these are all very superposition of one's own whims and fancies on molecules. Cytochromes, plastoquinone takes an electron from one point, one photosystem, and jumps grana to go to some other protein. Why? Why should it? It need not. So, when I presented this, if you are interested, you go to my YouTube, see what happens when I presented this in my talk with photosynthesis people. They start shouting, can't hear it. Because they have been habituated to that. But now you see how fast can a plastoquinone molecule go. Krishnamurti was saying, ions cannot move that fast, as fast, this distance. For example, from here, where your spine ends, the toe, until the whole thing, there is one neuron. It's a complete cell. A signal comes from that to here, through some ions and all that. Whole muscle contracts. How can you just calcium pumping and all that from here to here in a fraction of a second? All these are just coincidences. Sometimes elephants were seen there, Maruti was seen there. There is no physical correlation between the size, the speed, the ability. Some correlations, that's all. And mere correlations are not enough to get sound thermodynamic, mechanistic, kinetic, probabilistic logic. Number correlation is not enough. Alright? You need to make thermodynamic sense. So now I will go to the next part. I'll go quickly and then we'll have some interaction with students, primarily because we had promised that. So here is idea. One simple thing is enough to throw away Z-scheme. Yes. Sorry, number. Online or yes. Hello. Yeah. Sorry about that. You are right. Sir, what was the most fundamental, elementary thing? Anybody? Z-scheme is a sequential serial connectivity. Will it justify? Will it justify? No. Only parallel connectivity can justify enhanced oxygen production. Alright? Things like that. Anyway. So you people should say, now, when I was in Mala, I was a teacher in Mala, there was a girl in Rajgopal's class. Where is Rajgopal? Yes. Nita. One day we were sitting and discussing, and she came to me and said, Sir, ಇದಕ್ಕೆ ಗಣನ ಮಾಡಕ್ಕೂ ಇದು ಪಡೆದು ಮುಟ್ಟು ಪೋನು ಅವಳನ್ನು ಮುಟ್ಟು ವರಿನು ಇಂದು ಇದು ವೆಳ್ಳರಿಕ ಪಟ್ಟಣ ಪೋಲೆಯಾಣല്ലോ ಒಂದು. Exactly, that's how I felt when I studied that. Exactly. That's how I felt. So I thought all this had to go. I sat down and studied, came up with a better explanation. Alright? So, if you have genuine doubts, learn to, because that girl also came and said to me, I also realized, yeah, if I also felt, she also felt, it is not commonsensical. And then this logic, two things give greater current or electron flow, it can only be possible with parallel connectivity. Simple things. So like that. Now let me take you to the next part. The speakers of these talks were somebody different. The one guy has gone. Now, have you seen this bacterial flagella problem? You know that bacteria have flagella? So, now, the classical theory says bacterial flagella rotate. Rotate, this is the structure, alright? I have some video, a video in the Kadikyo. I have one section of the bacterial flagella, and there are only two rotary structures known. Both rotary structures our work has completely demolished. There was a scientist called Michael Behe. He said that rotary structures are irreducibly complex. In 1980s, he wrote a book. Why? To make this protein work, 7 distinct phases this thing will have to go through. It has to pierce. Have you seen any rotary structures auto-assembling? Any rotary structures? No. To get something rotate, say for example a fan, there is bush, all that, the assembly, it's an electromagnetic system where you can have bushes and rings and all that. But those kind of things to get in a natural system, it's impossible. Impossible. So this is the structure. You have a huge one, multiple protein here, and then there is a rod-like thing that goes out. So this is the dimension. When you study biology, please study dimensions, see if the dimensions fit, see if the forces agree. All those kinds of physical feel is very essential if you are studying biology. Otherwise you will end up saying 4 elephants can sit in a Maruti car. That's what. Alright? So you are supposed to know the dimensions and the forces at very sub-micron kind of level. That's what biologists are supposed to do. If you don't do that, it's very difficult. So rotation, it's very difficult. And this is supposed to rotate counterclockwise as well as clockwise. So it's supposed to be like this. If this is the piercing rod through the membranes and cell wall, this rotates at around, this big one rotates around 2000 RPM. 2000 RPM. If this rotates up at 2000 RPM, earlier this was supposed to be the primary rotor, but then they found that the idea was proton based, so when they mutated and found that nothing happens, then they said there was another MOT-B protein here. They said this rotates. So if this rotates at 2000, by the principles of gearing, this should rotate something around 13,000 or something, that high number. So not only rotating that number, it can switch from clockwise to counterclockwise. And that was through some way of gear switching. Such sophisticated materials and mechanisms in bacteria were advocated. This is the structure. This is flea-N, flea-M, flea-G. The name, this is not AB, flea-F. And this is the rod. This is the export apparatus. So if the flagella is made of flagellins, monomers, it's made of one monomer. The monomer goes through this and gets assembled. So there is the cytoplasm, cell membrane, periplasm, external barrier. Alright, so this is the rotor and this is the motor, stator earlier. But now this has become also a motor. Earlier they changed it. Okay, now I wanted to show you that. Can you? There's a video online, a 2017 Fluid Dynamics conference by yes, please see, this doesn't have any audio. See this. Many bacteria propel themselves by rotating thin filaments like this. This is what it is, and this is about a micron dimension. This is what you can see microscopically. Okay? Now, the flagella are approximately 20 nanometer in thickness. So the light that you can use, the lasers that you can use to visualize through fluorescence and all that, they are 200, or maximum resolution that you can get is 100 to 50 nanometers. The dimension is 20 nanometer across. So this rotation you can never see. Okay? So now what they did is, this flagellin they disassembled and assembled in lab. And then they stuck it on a film. And when the water flowed, you can see how that spiral flagella, now you see the water is flowing like this. It looks like rotating. Is that possible? If it is stuck at one end, can it rotate? And this doesn't have the basal assembly. Now this is just stuck. It doesn't have the basal assembly of rotor and motor and all that. This is just the filament. the filament is spiral. If it is stuck on the slide, the more you get, it looks like rotating. See all that. There is no motor, there is no rotor, it's just stuck. The material, when the water flows, it looks like it is rotating. Did you understand this part? So clearly, it is not rotation. We piece together that it is a precession. What is a precession? Now, if the water comes out here, I will show you what our theory is. That's enough. That's enough. Thank you. Let carry on. Put the other slide back, please. Thank you. Now, for me, if I was in the field, if I saw this, I would immediately throw out the rotary model. Because it's an optical illusion. Very clear that it's an optical illusion. I have shown you so many experimental observation, basic criteria, which a 10th class, 12th class student could have seen, questioned, and used to overthrow. So rotation, revolution, spiral movement, torsional, all these are very different. So if a rod has to move, advection, direct torque, gearing belting, these are the different ones. The natural way of, without having any problem of continuous 360 rotation, is only possible by human-made magnetic field incorporating systems. Mechanical systems, very difficult like that. So here is one beautiful thing. They are all type 3 secreting systems, the bacterial flagellar system. What is a secreting system? Something that pushes some internal component outward. So you see, type 3, type 4, this is the secretory system, and it is used to push some internal fluid out, toxins out, all that. So, in the murburn model, yet another aspect, it is a secretory system, it is not a rotary system. Now, yet another important thing, Howard Berg from Caltech, Harvard, and he was in these two institutes, he found this so-called rotation, and he said, he identified the earlier structures, said that it's a left-hand spiral. Now, if you take a left-hand spiral, and they said that it's a counterclockwise movement. If a left-hand spiral goes through a counterclockwise movement, it can, now this is the bacterium, if this is the flagellum, if this is a left-hand spiral, then if it goes through counterclockwise movement, then it will pull the bacterium through normal hydrodynamic basics, and it will not push. So anyway, the basic argument that they use cannot work. The most important thing is, this is a very important biophysics problem, because it's a low Reynolds number, something that he was talking about. At this dimension, the inertial forces don't work. Here, you have to think of some other way, So that's why they had these big ideas. And then the mechanism of bacterial flagellar motor was not based on ATP, but it was based on proton gradient. Somehow, the protons were supposed to come in like this. Protons came in, they set this going round and round. These kuti things set this thing round and round, and this long thing went round and round. So propelled like that. Now, the point is, and then uncouplers, they said that uncouplers were the reasons that showed protons were very important. Now, the problem is there are many molecules that don't have protons, but they also uncouple, so as to speak of. How? So it's not proton based. So if a proton can't go up and down a membrane on its own, something charged, nitro, 1 plus, 1 minus, plus, minus, these kind of molecules, how can they go like this? Just because some people said, they bought it. Proton is a very small unit charge. If it cannot go from here to there, how can these molecules with multiple charges, how can they go so quickly? That's another basic. What happened? Sorry. Yeah. Nothing. So, you see, this is the structure of bacterial flagella. Now, you see, they have grooves from the base connecting to outside, like this. The structure is like this, very beautiful. And not that is prokaryote, that is ordinary bacteria. This is archaebacteria. They also have these grooves, kind of. You see. Now, this is the classical rotary model, which says left-hand screw. We say this is not possible. The murburn model says metabolic water is pushed out and it goes through the grooves, and this one behaves like a elasto-hydrodynamic system. This filament is elastic and it vibrates, and that vibration passes on to the remaining part, and the one it looks like going around. Now it can explain how you stick it one place without rotor, without motor. So it's an optical illusion, this round going round and round. So there is this was an earlier slide. It's not actually a thrust. It is, now you imagine if water is just thrown out, it doesn't work. So I figured out that this part is elastohydrodynamics. Okay, so then you communicate. Sorry. The murburn model does not have both counterclockwise and clockwise. It has only counterclockwise. It doesn't say this. We say the apparent rotation is a optical illusion, and there is only counterclockwise precision that is afforded. So here you see the molecule, here if you see they have high amounts of polar and positively charged residues. So they can easily convert Ross into water and also recruit water there. So one very big thing is bacteria can even swim at pH 11-12 alkaline phase. So where is the proton there? So what they said, it's not proton there, it is sodium. Now you imagine where is one proton, what is one sodium? Very different ions. Like I said earlier, proton is 0.00002 angstroms, whereas sodium is 2 angstroms or more. Alright? So how can the same machinery, and now you see the structures. Oh, these are the structures here. The structures are not given here in this slide, I think. I put it together in the last moment. I'm sorry for that. The biggest farce is they said there is a one amino acid D32 conserved in this MOT. This is the MOT. There is MOT AB. I should have put the bigger image. This doesn't have it. So what happened is that is conserved. Now Danny checked the conservation and saw D32 is conserved. We found so many examples where there are no D or E. There are no D or E conservation. So what were all these people who are studying this field doing? You would ask. Whoever has money, salute. That's what they were doing. We have explored 20-25 different systems. We feel, oh, this is an interesting problem, we come there. There are many researchers working on this field for decades altogether. If they say this is conserved, they should have seen the conservation. They didn't. Somebody said it is conserved, so then it's not conserved. And there are many systems where there are multiple acidic residues. Then, if this is such a beautiful mechanically conserved system with gears and levers, you would imagine that their structures, the number of amino acids, all that will be also similar. There is no conservation whatsoever. The lengths, the types, everything is different. So, classical model of bacterial flagellar motility has proton motive force, MOT-AB as status rotating the serine, rigid rotating flagellum, propeller-like swimming. It uses proton motive force, the rotor hook, rotating filament, and swimming. Questions? Not enough protons. One mitochondria, it's just like a mitochondria. Bacterial body does not have enough protons. Then million type RPM, nanomotor, how can that be possible? Direct proof, there is no direct proof. No unified explanation of motility. Energetically not possible. So, what murburn model says is that when you do murburn for one molecule of glucose with murburn kind of oxidation, you get 36 molecules of water formed. If you study, analyze. So, metabolism produces lot of water. So, the water is extruded from that base of that flagella, and that creates a shear kind of force. It creates a precessing wave, and that by the phenomena of electrohydrodynamics, it gives thrust. Alright? Metabolic water generated by redox chemistry drives shear induced bending, no rotary motor. Alright? So this is the basic logic. So classical is PMF, rigid rotation, ion gradient, propeller flagella. This is metabolic water, processing, bending, metabolism, electrohydrodynamics, multiple motilities. It can explain very many different types. I'm winding up, sir. Don't worry. 4 o'clock.
Then we have to get from the new one also.
And you, yes. So classical proton-powered rotary motor, murburn redox metabolism produces water. So once again, so then biodiversity, ecology, dynamics. Now, why do we produce diversity? Fundamentally, when you have reactive species spontaneously interacting. So murburn is at a core bottom level, building blocks. They interact like electrostatic, reactive species, whatever. They give catalysis, conservation, and then from there central dogma and cell theory came. Then you have aerobic and anaerobic systems. So from chaos through time you can get deterministic order before affinity based mandates could set in. So foundation, this is the foundation of spectra of genes and species and ecosystems. Murburn allows distributed redox charge dynamics, energetic decentralization, microenvironmental niches. We got a work done which explains why microaerobic systems have greater diversity, because different redox regimes allow different kinds of metabolic stabilizations. So that's the reason. So molecular diversity from safe randomness, ecosystem level of integration. Then biodiversity, traditional explain is primarily through random genetic mutations. Add stochastic biochemical variations from redox reactions. Mutations are sponsored by murburn also. Slow over generations. Here within a generation also you can have rapid changes. Then roles of genetic DNA is a direct blueprint. DNA sets the stage, but murburn dynamics influence the real-time performance of the phenotype. Level of action, always focusing on genes and proteins. System level focus comes from murburn. So the biodiversity is an emergent property of redox flexibility and stochastic control in biological energy transduction. It transforms the understanding of the biodiversity from being a byproduct of random evolution to being a necessity or necessary obvious feature of murburn powered life. So, lignocellulosics, their breakdown, an enzyme cannot go into such complex structures. Only diffusible radical species can go and break down. So, carbon cycling, the natural formed halogenics, chloroperoxidase, 95% of naturally formed halogenics before mankind came, it was by enzyme action featured by mosaics. So, competition, symbiosis, succession, nutrient cycling, systemic health, everything so enables flexible metabolism and energy transmission at a micro scale, at a macro scale, links all living and non-living systems via diffusible redox couplings. Ecological scaling explains nutrient cycling, adaptability, dynamic equilibrium. Planetary scales describes as self-organized, non-equilibrium energy network sustaining life. Ecology is the emergent behavior of countless murburn interactions distributed across scales, from molecules to biospheres. So this is the kind of idea that I wanted to give you. Basically, at multiple levels, you have murburn working. It explains why you have DNA structured like this, why the bases are tucked in, and you have ribose and phosphate backbone on sites. You want to save it, that hydroxy groups and phosphate groups repel ROS, negatively charged oxygen with oxygen. So that way you can explain. Lipids, in the lipids you have multiple sessions. What is the most salient feature of life? A living being has the ability to preserve itself by commissioning non-spontaneous reactions. This is a physical perspective. What is the most essential requisite for the above? A simple powering modality interconnected with coherence, homeostasis, and response principles. Does the cell theory or central dogma provide a tangible means for achieving the above non-intelligently and in short time states? No. Murburn concept addresses the above need and complements the cell theory and central dogma as yet another fundamental principle of life. So what is the etiology expansion of Murburn concept? I hope all of you know. Mured burning or molecule unbound ion radical interactions or mured mild unrestricted redox catalysis. How is Murburn concept relevant to life? Murburn concept conceives cell as a simple chemical engine performing useful work via the principle of effective charge separation brought about by the obligatory involvement of diffusible reactive species or multiple things that Alan expanded perhaps. From the interactive chemo-electromagnetic matrix, so it sets in a matrix, electrostatic, diffusible species, all that, it sets in a matrix. Movement of one thing here perturbs the other. So that's how everything gets connected. And from that, emergence of biological intelligence. It's not that a membrane protein can pump 3 sodium out, pump 2 potassium in. All that intelligence can never happen. I can bet my life on it. It is impossible. And there is no evidence for it also. Why is Murburn concept a fundamental principle of life? Murburn concept provides a non-intelligent and stochastic framework of interactions between diverse molecules and ions that constitute the cell. In doing so, it enables a simple explanation for powering, homeostasis, coherence, signal transduction, electromagnetic activities, etc. So, last slide. What is the salient difference between classical and Murburn? Classical perceptions primarily deem DRS as toxic waste products, whereas Murburn school deems it as an essential elixir of life. Does Murburn concept oppose or negate classical perceptions? Murburn concept is a larger set of ideas, including several aspects of the classical views, incorporating later known facts and improving upon earlier prevailing stigmatic or aesthetic notions. Stigmatic and aesthetic notions mean, reactive oxygen species are bad, only bad. So in quantitative perspective, what is the impact of Murburn concept? Classical perceptions are purely deterministic schemes to account for metabolic or physiological outcomes, thereby mandating integral stoichiometry, whereas Murburn concept accommodates stochastic events, thereby explaining non-integral stoichiometry in cellular process as contributors of cellular function. So, with this, the formal lectures come to an end. It's already 4 o'clock. The organizers want us to end quickly, and the crowd has also thinned out.
Online.
Yeah, online. Yeah, more Can you? Mr. Mo, are you there? Oh, he has left. Yeah, he has left. So he has left. So no problem. So anybody wants to say anything at this juncture? Chairs, please.
Yes, that is the mechanism of bacterial flagella motor as well as the photosynthesis in a scientific way, but I think more studies are going to establish the fact as a whole.
Yes, madam. From 2007, we have just communicated that frontiers. I have four theoretically wrong ideas, 4 experimentally wrong interpreted ideas, and 4, what was the other aspect? 4, I gave you that yesterday. Anyway, there were 4. If you can take it, madam, and give me even refutation to one idea, experimental observations discounted, all that. Basically what has happened is a consensus has prevailed. Daniel Arnon, who, if you remember, cyclic photosynthesis, acyclic photosynthesis, those reactions. When he differed with the mainstream and he said that, oh, NADPH can be formed even at photosystem 1, it need not go through the Z scheme, they silenced him. Bicarbonate, very clearly the data clearly refutes photosynthesis, Z scheme. So, if you care, madam, my talk is available on YouTube. It elaborates, and we have published around 10 papers on photosynthesis. So it has systematically studied 2 papers published in JBSD. Systematically, all published data we have accounted for. We have refuted 45 points to say why the classical explanation is wrong. So in my philosophy, I can accept that murburn model can be wrong. But I will assert, let this be wrong, but I will assert that Z scheme is definitely wrong. Why? The explanation for the Emerson experiment, to explain higher yield, how can you put it in series? That's evidently wrong. Things like that, So things that don't, the 20% you get ATP with just rhodopsin. How can rhodopsin give? So rhodopsin can make reactive oxygen species. It's not a proton pump. Rhodopsin has got carotenoid. It is well-known chemistry. When carotenoid is hit by light, it produces reactive oxygen species, ROS. So I have been doing this work for about 27, 28 years now. I have conclusively shown that the classical theory, what we teach in school and colleges and in universities, is wrong without a doubt. Without a doubt, madam. Without a doubt. I can bet my life on it. It is wrong. I can also accept that murburn is wrong. Because there could be something wrong. But for sure, as of now, I don't know what is the wrong thing about murburn model. If you say this is not right about murburn model, and if it appeals me, I'll jettison it that moment itself. But we have got enough arguments and evidence, theoretical, experimental, all that, to show that the classical Z scheme is wrong. Koch-Jolio cycle is wrong, Q cycle is wrong. These are all part of the redundant idea. It is published, that material. In fact, my preprint assessing Z scheme as an explanation, it has got around 5,000 odd downloads in Open Science Foundation preprint, madam. And now I used to put my preprints there. They closed that preprint also for life science only. So now we have to go through all this. Only now I have ArcSiv and BioArcSiv and some other journals. So I was getting there were followers for my work, and that's a modality for them to disconnect me from my followers. So my paper in Biochemistry Insights, 10,000 downloads, most widely read paper. They discontinued that journal. So what to do? So I am very sure that the classical theory is wrong, but my theory also could be wrong. If you find some fault, I will be very happy. It will help me go to the right one. Yes, madam. Thank you.
Okay, all the way best for the.
Thank you, madam.
Yes.
Thank you, madam.
Thank you very much for the very good information about the. Thank you. It was an excellent, for me, new information about this concept.
Thank
For
you.
me to take some more time to accept this idea.
I wanted to share one video, if possible, to see the response. Yes.
Wait, how old is the new hire?
I don't know, but he literally.
Something Kalidas is something I forget, I am not very well versed, that he sat on the tree trunk and cut himself so he falls. So I don't want you to fall. I want you to be successful. But you please listen to reason, way of presenting, the way of writing. If you constantly attack, you are attacking yourself by doing that.
That's Andrew.
This is something that now you should move from the presentation society.
Please listen to it carefully.
For the past 4 years, my students barely published this. So now it is not published.
Yeah, but this is not the forum. Give a seminar to say, well, of course everybody knows that you
Now
know.
you know what until 4 years back this is
So, why did I do that? Because you keep on saying that this work is controversial. The last
Of course it is controversial.
Why is
It
it
is
controversial?
controversial because of you. I'm telling you. Because you don't know how to present it. You don't know how to write it. You don't know how to do it. You have to use simple, I even read your abstract. 10 words are there. So you want those people who know top English, they're bombastic English, they don't understand. Not as simple-minded things so that the common man publish it in Nature. And your work is that it should be published there. But you are not working in that end, you are working opposite. You are killing yourself by not listening to see how you can present your talk, how you can write your papers, so that people will understand your theory. I'm sorry, as I said, take my PhD, take my professorship away. Because I mean well. I don't know, you didn't say that, I'm saying that. I mean well for you, and I believe in you. That's why I wanted you to be here. But you disappointed me by not explaining your theory from the step one. I've never ever said that in my life to anyone.
I think it's a matter of India we don't question national something that you research and spoke for
As I said, I've taught for 40 years and I've listened to speeches after speeches and talks after talks since 1956. Two Marcus who came to our I know, I'm trying to ask because he did not bother to explain the concept. I've given you the papers. No, that's another thing. That is again very wrong for you to say. I've given the paper, Latha, make you read every word and you should understand. No, you should be able to write in a way that people understand. A man can say, don't explain because this is a problem. And they can deal with the problem. So unless you learn that you have a brilliant idea and you have to learn to sit down to write it in a way that people will understand step by step.
So, the thing is, it's not a tangent which.
No, but let me tell me that. I have presented it so
The thing is that we should not be aggressive about the tangent.
you are aggressive to remind you that your work.
This is the important place, huh? This is the very important place. This part is, yes. Yeah. What happened? Stuck?
In a way that people will understand step by step.
It's not a tangent which.
No, let me tell me that. I have presented it so
The thing is that we should not be
learn. And if you don't learn, well, you're wrong.
Yes.
Yeah, no, you are very, what's the next one? Arrogant.
Yes.
The arrogance at the highest level. No. I'm only interested in your life, only interested, I believe in you. Everybody says, Govinji, are you crazy? You're going to listen to what is talk? No. Because I thought I'd come here and it would explode.
So what does it say? It says, they know my work. They know my work. He says, are you crazy? You're going to listen to Murli's work. The work is published. It's out there. They pretend that they don't know it. Generations after generations, we teach wrong things. That's not right. When Newton's corpuscular theory was very popular, Huygens proposed the wavefront theory. It took 200 years almost. 200 years. Just because there is somebody powerful, why should people not listen to arguments and evidence? Science is based in claims, arguments, evidence. Claims, arguments, evidence. That's it. Believe in it, young kids. You should do that. We have one more professor, what's his name? K.V. Venkatesh, star professor from IIT Mumbai. He is supposed to join in. I hope he will join. He and Dr. Grinson, they are the valedictory guests. So, are we going to valedictory now, madam? Sir?
Yeah, we will continue.
We'll continue? So, can I just call him? I said around 5 o'clock, it's only 4:20 now. Yes, okay, sir.